Good morning, my dear students. Welcome to our zoology online class. Dear students, the last class we studied about two important topics. The first topic is RNA word, and the second topic is properties of genetic material. Okay, so in the last class we studied about the RNA word. This is very important 5-mark question, and the next 5-mark question is the properties of genetic material. Okay, so dear students, hope you all studied these two question answers. Today we are going to study about some more topic. So listen very carefully, my dear students. So the first topic of today's class is packaging of DNA. Okay, packaging of DNA. So in this topic we are going to study about the structure of DNA molecule. Okay, so listen very carefully. In this topic we are going to study about the structure of DNA molecules. Okay, dear students. The first point about this DNA molecule is, already in the last class I told you, the DNA molecules are made up of base pairs. Okay, nitrogenous base pair is one of the important property of the genetic material. Okay, the adenine, guanine, cytosine, thymine, these are the nitrogenous base. Okay, so the first point about this DNA is the distance between two nitrogenous base pair is 0.04 nanometer. Okay, 0.04 nanometer is the distance between two nitrogenous base. Okay, two nitrogenous base or base pair. Understand of you? Then, second point, to find out the length of a DNA molecule, what formula is there? Okay, so if you want to find the length of a DNA molecule in a cell, the number of base pair present in the DNA is multiplied with the distance between the two base pairs. Okay, so to find out the length of a DNA molecule, we have to multiply the number of base pair with this distance between two base pairs. Understand of you? So now we studied two important points about the DNA. The first point is the distance between two base pair is 0.34 nanometers, the first point. And the second point, to find out the length of a DNA molecule, the number of base pair is multiplied with the distance between two base pairs. Okay, so it's the second point. The third point, in Escherichia coli bacteria, if the length of the DNA in Escherichia coli bacteria is 1.36 micrometer, means the number of base pair present in that DNA is 4 into 10 raised to 6 meters. Okay, 4 into 10 raised to 6 meters. Listen very carefully. Suppose in Escherichia coli bacteria, the length of the DNA molecule is 1.36 micrometer, means the number of base pair present in that DNA is 4 into 10 raised to 10 meter. Understand of you? So hope you are understood this third point. So now we have completed 3 points about the packaging of DNA. Then next, one scientist, his name is Duprat. Okay, his name is Duprat. See, he only first proposed the single strand model of a DNA molecule. Okay, he only first proposed the single strand. Actually, DNA is a double stranded molecule. Here, this person only first introduced or proposed the single strand model of a DNA molecule. And here, the single strand DNA molecule is closely associated with a protein. Name of the protein is called histone protein. Name of the protein is called histone protein. So once again, repeat this point. Duprat only first proposed the single strand model for a DNA molecule. And according to his model, the DNA molecule, single stranded DNA molecule is closely associated with a protein. Name of the protein is called histone protein. Okay, then the next point, actually in plants and animals, okay, plant species and animal species, the number of DNA molecule is more than the bacterial cells. Okay, in plants and animals, the number of DNA molecule is very high than the bacterial cells. Okay, so all these excessive, the large number of DNA molecules are folded, then only it can fit inside the chromosome. Okay, then only it can fit inside the nucleus. Understand you? So hope you understand this point. In plants and animals, the number of DNA is very high than the bacterial cell. So all the DNA molecules in plants and animals, the DNA molecules are folded many times. Then only it can fit inside the nucleus. Okay. Then next point. In Escherichia coli, there is no nucleus. Okay, there is no nucleus. So, the DNA molecule in Escherichia coli bacteria, the DNA molecule is held with a protein molecule in a region. Name of that region is called nucleus. So, inside the bacteria, Escherichia coli bacteria, one part is one region is there. This region is called nuclear region. In this nuclear regions only, the DNA is held with a protein molecule, because in Escherichia coli bacteria there is no nucleus. This is a reason. Okay, so because of the absence of nucleus in Escherichia coli bacteria, in Escherichia coli bacteria, the DNA is held with a protein in the nuclear region. Nuclear region. Understand, my dear students? Yes, students, now we are going to study about one of the very important two-mark question. Genophore. Name of the question is genophore. Here, genophore means maximum in prokaryotic organism, the DNA is circular in shape. The DNA is circular in shape and the chromatin fibers are absent. Okay, chromatin fibers are absent. So this condition is called genophore. This condition is called genophore. So once again repeat the meaning of this word genophore. In prokaryotic organism, the nucleic acid, the DNA is circular in shape. Okay, circular in shape and the chromatin fibers also absent. So this condition is called genophore. Hope you have understood this point. Then next point about this DNA is in eukaryotic organism, the chromatin fiber is made up of many repeated units, many repeated subunits. Name of that unit is called nucleosome. Okay, listen very carefully. In eukaryotic organism, the cells consist of many chromatin fibers, and these chromatin fibers are made up of a repeated subunit or repeated unit. That repeated unit's name is called nucleosome. That repeated unit name is called nucleosome. Then one scientist, his name is Kohlberg. Okay, his name is Kohlberg. He only first proposed the structure of this nucleosome. He only first introduced the structure of this nucleosome. So according to Kohlberg's opinion, the nucleosome model is made up of two molecules of 4 histone proteins. Two molecules of 4 histone proteins. So a single histone molecule, a single nucleosome molecule is made up of 8 histone proteins. 8 histone proteins. So listen very carefully. I repeat this point. Kohlberg first proposed a model for the nucleosome. According to his model, a nucleosome is made up of 2 molecules of 4 histone proteins. Okay, so totally 8 histone proteins are present in a nucleosome. Okay, so it is called histone atomer. This is called a histone atomer. Understand of you? Understand? Then actually the DNA molecule, the next point about this DNA is a negatively charged molecule. DNA is a negatively charged molecule. This negatively charged DNA molecule is always present or around the outer surface of the nucleosome. See, just imagine it's a nucleosome. Okay, so the nucleosome, inside the nucleosome, just imagine it's a nucleosome. It is a positively charged molecule. Nucleosome is a positively charged molecule. So here, the DNA is a negatively charged molecule. Okay, so the negatively charged DNA molecule is 2 times warp around this nucleosome. Okay, the outer surface of the nucleosome only the DNA molecule is present. So the nucleosome is surrounded by the DNA molecule, and the DNA molecule is a negatively charged molecule, and 2 times it is surrounded. Okay, the DNA molecule is surrounding 2 times in a single nucleosome. Hope you are this point. Okay, hope you are understood this point. So DNA is a negatively charged molecule. This molecule is 2 times or 2 times it is surrounding the outer surface of the nucleosome. Understand of you? Understand of you? Then in some nucleosome, okay, see, in some nucleus, the nucleosome, the 2 neighboring or 2 adjacent nucleosomes are linked with a DNA. This DNA is called linker DNA. Okay, this DNA is called linker DNA, and this linker DNA is otherwise called H1 DNA. The linker DNA is otherwise called H1 DNA. Listen very carefully. The nucleosome. So just imagine this is one nucleosome. This is the other nucleosome. These two are the neighboring nucleosomes. Okay, these two nucleosomes are connected by another one DNA. That DNA name is called linker DNA, otherwise called H1 DNA. Otherwise called H1 DNA. Okay, understand the next. Listen the next point very carefully. In some nucleus, the linker DNA or the H1 DNA is absent. Okay, the linker DNA or the H1 DNA is absent. Okay, so in these type of DNAs, the DNA is arranged as a beads on a string appearance. Beads on a string appearance. So just to imagine the linker protein is absent here. Okay, sorry, the linker DNA is absent in this nucleus. Okay, in some nucleus. So the nucleosomes are arranged as a beads on string appearance. Beads on string appearance means, so just imagine this is one nucleosome, this is another nucleosome, this is another nucleosome. So the DNA is entering inside the nucleosome through one region and coming out through another one region. Okay. Then it is again entering into through one region of the next nucleosome and coming outside, and again entering and coming outside. Okay. So this appearance is what appearance? Look like a beads on string appearance. This appearance is called a beads on string appearance. So hope you understood this point. So in some nucleus, the linker DNA or the H1 DNA is absent. So in this condition, the nucleosomes are arranged in a beads on a string appearance. Okay, beads on a string appearance. That means the new DNA is entering through one region of nucleosome and passing to the other region from the nucleosomes. Understand? Understand? I hope you understood this point. Then the next point is, say, actually, the chromatin fiber in the interface nuclei. Okay, the chromatin fiber in interface nuclei and the mitotic chromosomes. Okay, chromatin fiber in the interface nuclei and the mitotic chromosomes. So in these chromosomes, they have a diameter. Okay, they have a size. Okay, so this diameter is vary from 200 to 300 nanometer. Okay, 200 to 300 nanometers. So once I repeat this point, see, chromatin fibers in interface nuclei and the mitotic chromosomes have a diameter. That diameter is vary from 200 to 300 nanometer. Okay, 200 to 300 nanometer. Understand? Then the next point is 30 fibers are arising from the folding of a nucleosome. Okay, 30 fibers, one more question. 30 fibers are arising from the folding of a nucleosome. Understand a few? Then it is changed into another one structure. Name of the structure is called solenoid structure. Name of the structure is called solenoid structure. And this solenoid structure consists of 6 nucleosomes. This solenoid structure consists of 6 nucleosomes. So once again repeat this point. 30 fibers arising from the folding of the nucleosome, okay, into, and it is converting or it is forming another one structure. It is called solenoid structure. The solenoid structure consists of 6 nucleosomes. Hope you understood this point. Then the next point is, say, maximum the DNA molecule, okay, consists of 40 foldings. Okay, in the ordinary DNA molecules consist of 40 foldings. Understand a few? Then to keep or to pack all the chromatin fibers, the DNA molecules need some additional set of proteins. The DNA molecules need some additional set of proteins. That additional set of proteins are non-histone chromosome proteins. Non-histone chromosome proteins. So once I repeat this point. To pack all the chromatin fibers, the DNA molecules require or need some additional set of proteins. That proteins are not a histone protein. That proteins are called non-histone chromatin protein. Non-histone chromatin protein. Okay. The last point about this nucleus, this DNA is, in nucleus, some regions are loosely packed or loosely arranged. That regions are called euchromatin. That regions are called euchromatin. And some regions are tightly packed. That regions are called heterochromatin. And here, this Euchromatin is active in condition. Transcriptionally it is active, but the heterochromatin region is in inactive condition. Is in inactive condition. So once again repeat this last point. In nucleus, some regions of the chromatin fibers are loosely packed. Okay, that regions are called euchromatin, and that region is transcriptionally is active region. But some regions are tightly packed. Okay, some regions are tightly packed. So this region is called heterochromatin region, and these regions are inactive in condition. Understand, my dear students? So hope you all understood this first topic, packaging of DNA. So now we completed the packaging. What are the substance or what are the materials present in the DNA molecule, and length of the DNA molecule, everything here completed now. Okay? Then hope you all understood this topic. And the next topic is so dear students, now we are going to study the next topic. Okay, our next topic is DNA replication. Okay, DNA replication. Actually, this replication inside the cell, this DNA replication process takes place during the S phase stage. During the S phase stage. Okay, actually this replication is a process by which the number of nucleic acid is increased. The DNA molecules are increasing in their number. Okay, so during the replication process, the number of DNA molecule is increased, and this replication process takes place during the S phase stage of the cell. Okay, then actually the replication process is classified into three types. Okay, replication process is classified into three types. The first one is conservative replication. Okay, second one is called dispersive replication, and the third one is semi-conservative replication. So the first one is conservative replication, and the second one is dispersive replication, and the third one is semi conservative replication. Okay, so the replication process is divided into three types. The first type is called conservative replication, second type is called dispersive replication, and the third one is called semi-conservative replication. So now we are going to study about all these three types of replications methods. Okay, all these three types of replications. Okay, so the first one is conservative replication. Okay, see, in this type of replications, first the original double-strand DNA molecule is acting as a template strand for the production of new strand. So just to imagine, it's a double-stranded DNA molecule. This double-stranded DNA molecule acts as a parent DNA or acts as a template DNA. So this double-strand DNA molecule produce two daughter strands, okay, or two new DNA molecule. Okay, so this type of replication is called conservative replication. So conservative replication meaning, so in this type of replication, the original DNA molecule, double-strand molecule is acting as a template, and this template DNA molecule is synthesizing or producing new strands, new DNA molecules. Okay, then second one is called dispersive replication. In dispersive replication methods, just imagine it's a DNA molecule. This DNA molecule is divided into fragments. So just imagine this is one fragment, this is another one fragment. Okay, so here these two fragments are producing two new DNA molecule. Here the new DNA molecule consists of one old strand and one new strand. Okay, so a single double-stranded DNA molecule is divided into fragments, and each and every fragments are producing new DNA molecule. Here the newly produced DNA molecule consists of one old strand and a new strand. Hope you are understood this dispersive replication method. And third one is semi-conservative replication method. Here, the semi-conservative replication method was first introduced by two scientists, Watson and Crick. Okay, in the year of 1953, this Watson and Crick, these two people only explained the semi-conservative replications method. Okay, so just imagine, it's a DNA molecule. It's a double-stranded DNA molecule. Okay, so in this type of semi-conservative replication method, first from one end the DNA molecules are unbinding or separating. Okay, so actually this DNA is a double-stranded model, is a spring-shaped model. Okay, so from one end the two strands are separating. From one end the two strands of a double-stranded DNA molecule is separating. Okay, and the hydrogen bond present between these two strands also broken. Okay, so these two strands are connected by hydrogen bond. You all studied. Okay, already we have studied. So the hydrogen bond present between these two strands also broken. Okay, hydrogen bond present between these two strands also broken. So now in a single double-stranded molecules, one end is separated. Okay, now these two separated strands are acting as a template strand and they are finally producing new strands. Okay, these template strands producing new strands. Understand of you? So once again repeat this semi-conservative replication method. So in this type of replication method, this type of replication method was firstly explained by Watson and Crick in the year of 1953. Okay, so in this type of replication method, first one end is separated. Okay, one end of the double-stranded DNA molecule separated and the hydrogen bond present between these two strands also broken. Between these two strands also broken. Understand of you? And now these two strands are acting as a template strand and these template strands producing new strands. Understand, my dear students? So hope you all understood this point. Okay, these types of replication methods. Okay, then next class we will study about two important replications. Okay, say the experimental proof for replication process. Okay, this is very important 5-mark question. We are going to study in the next class. So today we have completed the packaging of DNA and just we started the types of replication method. 3 types of replication method: conservative replication method, dispersive and semi-conservative method. So this is very important for 3-mark question and packaging of DNA helix is a 5-mark question. Okay, in packaging of DNA there is many 1-mark question is there. For example, the distance between two base pairs. Okay, so and nucleosomes are made up of how many proteins, histone proteins. Like that, many 1-mark questions are there. So today itself, try to complete these 2 topics. Okay, students, we will meet in the next class. Thank you.